Good evening, everyone. My name is Shaya and I am a junior here at Holy Names. My research project is on the effects of menopause and high-fat diet on adult hippocampal neurogenesis in a mouse model of Alzheimer's disease. And I'm working with Dr. Charlie Abigahanam at Albany Medical College. In essence, I will be counting the amount of stem cells in each developmental stage in mouse brains with Alzheimer's disease after 10 months and how the amount differs in mice with menopause and simulated metabolic disease. There were 4 experimental groups. One group of mice was fed a low-fat diet and had induced menopause. Another was fed a low-fat diet and did not have menopause. One was fed a high-fat diet and had induced menopause. And the last was fed a high-fat diet and did not have menopause. Both metabolic disease and menopause are risk factors for Alzheimer's and play a role in the development of the disease. Metabolic disease was simulated by feeding the mice high-fat food, and the mice had menopause induced. To do this, the mice were injected with 4-vinyl cyclohexene dipoxide, or VCD, which kills primary follicles, stopping the release of any eggs. This method causes a period of estrogen fluctuation similar to human menopause. This is a better option than surgically removing the uterus, since that would result in a sudden loss of estrogen, which is unrealistic. This is a timeline of the experiment. Everything on this timeline already took place. The mice already had their tissues collected, and my role is to perform data collection and analysis. So as I said, I will be quantifying the effect of both menopause and metabolic disease on Alzheimer's mice brains by counting stem cells. Stem cells are undifferentiated cells, and when we are embryos, we have a lot of them, which as we grow will become all of the cells of our body. As adults, we don't have as many stem cells, but they still exist to replenish important cells, most notably in our bone marrow and in our brains. In the brain, new neurons are formed in a few places, including in the dentate gyrus of the hippocampus, which is where my study is going to be looking. Quiescent neural progenitors are a form of dormant stem cells in the dentate gyrus, and those are the green ones that look like little trees on the left. Amplifying neural progenitors are an active form of these stem cells in the dentate gyrus, and those are the flat ones in the middle. These cells will multiply, giving rise to either glial cells like astrocytes and oligodendrocytes, which are to the left, or granule cells, which are to the right. This process is known as adult hippocampal neurogenesis. In brains with Alzheimer's disease, adult hippocampal neurogenesis is decreased. My hypotheses are menopause will decrease adult hippocampal neurogenesis in a mouse model of Alzheimer's disease, and metabolic disease will decrease adult hippocampal neurogenesis in a mouse model of Alzheimer's disease. I'm going to determine the amount of adult hippocampal neurogenesis that occurs by counting the number of each type of stem cell and mature neurons. To make sure that we can tell the difference between new cells and old ones, we used 5-ethynyl-2-deoxyuridine, or EDU, labeling. EDU is a molecule that mimics thymine, a component of DNA. So any new cells that are born after EDU is added will have EDU in their DNA, which allows us to distinguish between the new ones and the old ones. In order to determine the difference between each cell type, we will be looking for specific proteins expressed by each. Double cortin, which is up there as DCX+, will be used to identify immature neurons. Neuronal nuclear antigen, which is up there as NEUN, will be used to identify granule cells. Oligodendrocyte transcription factor 2, OLIG2, will be used to identify oligodendrocytes, and glial fibrillary acidic protein, or GFAP, will be used to identify astrocytes. Finally, nestin labeling will be used to identify neural progenitors, which will be classified as active progenitors, the flat ones, or inactive progenitors, the trees, according to their morphology or their shape. Using this method allows us to not only see how many mature granule cells and glial cells were born meaning how much neurogenesis occurred, but also if the maturation of these cells got stuck anywhere. And this information gives us a window into how Alzheimer's disease prevents new neuron formation, meaning if the cells are simply stuck in an inactive state and never form, or they get stopped somewhere in their development. I've not performed this experiment yet, but in order to prove my hypothesis correct, the results would need to indicate that the group with induced menopause and fed a low-fat diet has a lower amount of amplified progenitor cells than the group without menopause and fed a low-fat diet, and the group without menopause and fed a high-fat diet to have a lower amount of amplified progenitor cells than the group without menopause and fed a low-fat diet. This is my bibliography. And I would like to thank Dr. Avi Gahanim for working with me, and Mr. Lakari and the Holy Name Science Department for all their help this year. Come back next year to see my results. And does anyone have any questions?